Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology Practicals. This is called Practical 9. Investigate the antimicrobial properties of plants. The plant chemicals or plant substances are going to be tested on microorganisms because these are antimicrobial substances. So we're going to be working with microorganisms, some of which could be pathogenic. So we have to make sure that we wear gloves. And again, this is to prevent maybe some allergens that will come from plant materials, as well as the microorganisms themselves infecting you or you contaminating the culture. We use aseptic conditions like flaming in order to prevent contamination or contaminating microorganisms. Make sure you do not work with pathogenic microorganisms because then you provide conditions for you to be infected. You also have to make sure we culture at temperatures that are below the body temperature, again to prevent the culture of pathogenic microorganisms. During the culture, we have to decide, are we going to carry out a liquid culture or a solid culture? And then we have to choose the optimum conditions, like temperature and pH. So the procedure is wash your hands with soap and water and disinfect the bench. Then place the material to be observed, like a piece of garlic, in the mortar and grind. This is an extraction method because, again, we're working with plant materials. So we have to find ways of extracting these antimicrobials from the plant materials. That means we need to grind to increase the surface area, and we need to use a suitable solvent in order to extract out as much of the antimicrobial chemicals or substances as possible. So using a mortar to grind, this is to increase the surface area to make the plant materials as thin as possible to expose more leakage or more substances to leak out. We're going to add 10 centimeters cubed of an alcohol to the garlic. The alcohol here is acting as a solvent to cause as much extraction of the antimicrobial substances as possible. Then we can use forceps to pick up the paper discs and place in the mortar to soak up the garlic and alcohol solution. The paper discs are going to be like filter paper discs that are cut to be the same diameter. These are going to be placed in the extract solution in order to get out the antimicrobial substances. And again, we can use forceps to place the discs on a sterile agar plate to dry. This is to ensure that the alcohol evaporates, leaving only the antimicrobial soaked in the filter paper. Now we're going to get a petri dish and we'll mark 4 quarters or 4 areas. For example, let me show you. If this is the petri dish, we're going to cut out to create 4 quarters. This could be the control and this could be concentration 1, concentration 2, concentration 3, depending, or it could be antimicrobial 1, antimicrobial 2, antimicrobial 3, depending on if the extracts are coming from 3 different plant materials. One could be the control. So like I said, use a marker pen to mark 4 quarters on the bottom of the agar plate that has been seeded with bacteria. There is already bacteria on this agar plate. The 4 quarters could include 3 test experiments and 1 control. You open the lid of the petri dish and now we're going to use forceps to place the specific filter paper soaked in the antimicrobial substances in the different area. So we could place one here, another here, and another here, then the control one here. While placing the soaked filter papers, we have to make sure it's done opening away from ourselves because, of course, we can have some microorganisms coming from our breaths going into the sample and contaminating it. Then we can close the petri dish, invert it, and then place it into an incubator to maintain the specific temperature. In carrying out this experiment, we have to make sure the pH is constant. You could do that using a buffer. The temperature has to be constant. We could use an incubator. And all the other variables have to be constant. The only difference is maybe we have different plant extracts. Everything else has to be the same. So we can culture for 24 hours and we use a ruler to measure the diameter of the zone of inhibition around each disc. We can see this is going to be the diameter. And then we can record the results in a table. Problems associated with this method of measuring the dependent variable is that sometimes the diameter is not going to be a perfect circle. So some results could be a little bit erroneous or there could be some tiny variations in the results. So this brings us to the end of this core practical. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.